So it's my great pleasure to introduce Arvind Kumar, who is Associate Professor of Computational Neuroscience at the CST Division in EECS. He is currently the coordinator of the Brain Health Research Initiative at KTH, and previously PI of D-Brain, which is how I got to know digital futures personally. So today, Arvind will let us know about brain disease and the digital futures. Please.
Thank you, Martina. Thank you for this opportunity. I remember when I agreed to give this talk. Last week, I was kind of sweating. Why did I agree? So, because I think I promised too much, at least to myself. Still, I think it will be, let's keep it an interactive session. There are some points where I would like to have your feedback. There are some controversial and provocative thoughts, so you should just attack me on that in response. And maybe I'll give you a little bit about what the future might hold and how we could change things. So a lot of people have helped me develop these ideas, particularly people who are in my group. And also now colleagues in the Brain Health Initiative. And in fact, this particular idea that I'm going to present today very much precipitated from discussions that happened in those meetings. So I'm really thankful for them, and particularly Janet, Eric, Thruff, and Uli, who actually helped me shape these thoughts a bit more. The person who is really responsible for this talk is Pascal, who actually happens to be in the audience. If I think he had done good work, we had clear results, this talk wouldn't exist. Since he didn't have good, clear results, we thought we have to defend him somehow. So this is the response, okay? All right, so let's start a little bit back in the past. So after my master's, I spent some time in one of the hospitals in India doing an internship, trying to change from electrical engineering to something else. And I worked in this building here. This is the NeuroLogic clinic there. And on the other side of this building was the cancer center. And those 6 months that I spent there, I saw two things. People came to this building. They were, subjected to many different tests. They went away without any improvements in their symptoms, but with a name, with a diagnosis. In this building, people entered and usually did not come back. So it was a very, very sad time. I mean, if you think about it, of course, I was young and, everything was looking good. We'll change the future. So I didn't care much at that time. But still, there was a situation. So in cancer, a lot of progress has happened. And you could attribute it to basic science, amount of funding and whatever, even though the number of incidences are increasing. We are actually improving quite a lot in terms of treatment, and in many cancers, people can actually have their full lifespan. What is in brain disorders case in the last 25 years? We still have this problem. A lot of healthcare costs is going towards brain disorders. Longevity has increased and is increasing, and that means incidences of neurodegenerative diseases will increase, and younger people, they are for whatever reasons, they are more vulnerable to psychiatric disorders. So overall, this is increasing. And in terms of treatment, what we have is this. So this is the Bible of a neurologist who consulted to diagnose the disorders. This has existed for many years, and this is how it has evolved. So it has gone from 130 pages to 947 pages, from 106 disorders to 410. And after that, in this one, they don't give how many disorders are there because they created more subclasses. So we have gotten amazingly good at diagnosing these disorders, classifying them. But in terms of treatment, I think it's still the same. And this is something that I want to think about a little bit in this next 40 minutes or so. So what is a brain? A brain is some material stuff which has its physics and chemistry, physiology, and dynamics. And on top of that emerges the mind, which is about finding invariances, solving problems, creating algorithms and regularities, and so on. But if we dig into the brain part, I think every one of us who belong to the neuroscience community, they have their favorite entry point into this problem. Some people study at the whole brain level. Some people like to think of the brain as a network of networks, and you can go down all the way to molecules. And in the process, we spend like 10, 12 orders of magnitude in both space and time. Now, this is an old picture, very nice, and thanks, Janet, for this, but a more updated version of this reads like this, because the picture below kind of gives you some hierarchy, which is definitely not true. There is some semblance to that, but really the picture is that all levels could affect other levels. Therefore, the brain functions or dysfunctions that emerge, they could start anywhere in this and manifest at multiple levels simultaneously. So this is something we should keep in mind. So now with this background, let's start to think about brain diseases. So how do we diagnose these? Usually the diagnosis starts with behavior. And I don't think I have to tell you what this person may have. Any guesses? So there are a lot of people who study basal ganglia, so no guesses needed. But there are other diseases where we basically rely on behavioral assessment. Sometimes we give them questionnaires, sometimes we ask them to do certain behavioral tasks, like in Parkinson's case. This is a nice example of stroke where you have this hemispatial neglect. I don't know this disease, what do you call it? Is it depression? Is it anxiety? And so on, right? So we usually start with some behavior. And once we have the behavior, then we follow a simple plan, at least in the classical neurology setting, that you have behavioral problem. There must be a cause for this. And where is that cause? That cause must be at the very core of brain's hardware. That is, it must be there in terms of brain's chemistry, its morphology, its genetics, and so on. And this legacy goes back to John Martin Charcot, who was, I think, maybe the first neurologist who started to classify patients. And his belief was that all of these physical symptoms must have a physical cause, a physiological cause. And therefore, he always tried to argue for this. And that basically set us on the path of what we call as biomarkers of the diseases. So whenever there is a behavioral problem, we want to know in terms of brain's hardware, brain's structure, its chemistry, what are the predictors, what are the determinants of a particular disease? Like for Alzheimer's, we look for tau protein. For Parkinson, we look for the disease damage in the, let's say, dopaminergic system, and so on and so forth. And what this has created is, I'm using this word deluge because this will become clear a bit later, but in recent times, what has happened is that because of this biomarker search, a lot of repositories have popped up. ADNI is there for Alzheimer's, PPMI for Parkinson's disease. And these repositories are full of data. They have brain images, they have gene markers, they have blood markers, protein expressions, physiological measurements, behavioral measurements, tons of data is there. EEG, you name a disease, and you'll find a repository, if not now, maybe in a month or so. So things are growing rapidly in this. And there's a huge amount of data. What do we do with this data? The typical approach is that you record some biomarker. Maybe you have a hope or expectation that this particular readout could be relevant. You measure it in a healthy cohort and in a patient cohort. And then the game used to be that find statistical differences. Let's say you are measuring tau protein. You look at the expression in Alzheimer brains, in normal brains, age matched, and so on. And if you see that there's a statistical difference between the two cohorts, then you say that, yeah, this is a biomarker of this disease. More recently, with the advent of machine learning, I think we are going away from this notion of p-values. We say that, can you classify? And in this case, it's a binary classification problem. And I personally think that that's much more relevant than finding these p-values. And I was looking for it, this idea of healthy controls. This actually is not that old. So when I looked into PubMed with keywords like Parkinson's disease and Parkinson's disease with healthy controls, you see this kind of readout. So there is an exponential growth in number of papers that are published with healthy controls. So it's a very, very popular approach to find biomarkers. And obviously, this is not so difficult to understand. I'm using this very old quotation from Francis Crick, who said that a person's mental activities are entirely due to the behavior of nerve cells, glia cells, the atoms, ions, and molecules that make them up and influence them. And if you go into lab setting, this is how exactly it works, that you can find causal links between, let's say, one part of the brain's hardware and some behavior. There's no confusion about it. So when I started to think about brain diseases for various reasons, I noticed that one thing that was not part of the brain disease diagnosis was brain's activity. And that was quite a surprise. After all these experiments, they are telling us that there's a causal link between brain's activity and its behavior. However, if you go to the clinic, except in the case of epilepsy, nobody cares for brain activity. And I was wondering why, because partly from survival reasons, that I know something about brain activity. I want to be relevant in this problem domain. So if we know that brain activity is actually linked to the disease states, then we have some theories that could explain it and maybe even suggest means to change it. Because if the hypothesis becomes that brain activity is the sole determinant of behaviors, then you can manipulate it. So with that, Pascal came along, D-Brain was funded, Digital Future supported us, and we said, okay, this is a nice opportunity to jump into this. So Pascal joined, and we asked Daniel. He is the head of the NutMeg, which is the national MEG facility in Sweden. And they have tons of data about brain's activity in different disease conditions, sometimes in healthy cohort doing function as well. And at that time, if you look at the data about Parkinson's disease, that was very sparse, but still there was some consensus emerging about interactions between different areas, which was going above the notion that Parkinson's disease at the electrical activity level is just beta. So Pascal came along and he said that we should analyze the data differently. And his idea was that let's look at the spectra, but not look at the peaks, but look at how the aperiodic activity is the spectrum, how it falls as a function of frequency, and that's this lambda. So that's how Pascal started his postdoctoral work here, and he analyzed this data. This is how lambda changes in one person over time, but then we said, okay, this is too much, less average, because there are too many brain areas to deal with. So this is what he did. Then he compared different brain regions in healthy cohort and the Parkinson's disease cohort on medication and off medication. And he found a lot of significant differences. So one big result was that when you compare Parkinson's patients without medication with healthy controls, the slope of aperiodic activity is significantly different. In Parkinson's patient, in the posterior regions, it's actually much higher than healthy controls. And look at the p-values, it's amazing actually. In some areas we have 10 to the power minus 3 or so. So that's pretty good. We also found some correlation with the age, specifically in the Parkinson's patients. And this was quite encouraging, let's say, at that time. And because mainly that we found out that there's this unexpected change in the slope, and this we interpreted as a balance of excitation and inhibition. And more important, I think at that point we thought was that this particular analysis brings sensory areas in the focus, because this is something that people don't think about the disease. We did not find any effect of medication on this parameter. We didn't find any effect of the motor behavior and so on. However, we thought that this could still be relevant. Maybe you can plan your DBS surgeries using the idea of EI balance, also corroborated by many other theories. But was it actually really useful? I mean, could I go with that data to a doctor, to Per Svenningsson, and say that, Per, now stop all your blood biomarkers. I have this thing. Put the people in scanner for 5 minutes, and I'll tell you if they have disease or not. No way we can do that. Despite all these amazing significant values, if we try to classify patients based on this measure, there's no chance. Too many false positives, false negatives. But we wrote the paper, it got published, it gets cited, everything. So what is happening? So we look at other data who follow similar approach. They report significant values, and Usually they don't report this, but when they do report F1 score, meaning your classification accuracy, precision, and recall, etc., usually it ends up in this range. And this is, I'm talking about for binary classification. This is not good for clinical practice. And I think earlier this year we had a talk here by Josephine who explained it, that this is useless for clinical. This may be good for giving you some lead into the disease, but clinically not. But when it doesn't work, people like me and others who analyze these cohorts, they say, yeah, we had only 18 patients and 18 controls. If I had 18 million, it would be good because I'm perhaps overfitting here and so on. So this is a typical thing that is argued. But why is it not working? Let's step back and think, why is it not working? Let's believe that these observations are right. What could be the reason? Any guesses? Do you want to take any guess why it is not working? So if you push people that, wait, it's not a data problem, what else could it be? Well, maybe the data is not of good quality. It's MEG data. There could be artifacts. People are not doing a task here. They are resting state. Or it's fMRI. It doesn't have enough time resolution. So we come up with this reason. Or we say that this was just a trial, right? We were searching for maybe MEG-based biomarkers. Maybe this is not. Maybe there will be something else. So this is fine, acceptable. Could also say that our algorithms were not good, and in some sense this is true, that conventionally you were doing spectral analysis. Now we can do more sophisticated machine learning things where we don't know what is happening, but we hope for the best. So you could argue all these things. However, all these arguments eventually come down to this, but give me more data. I promise it will work. But maybe this is not the case. Maybe there is a problem, and this is perhaps the most provocative part of this talk now. So let's just think about this. You go to your hotel room, you put in your card, you see the switches. Can you actually tell if the light is on or not in the main hall of your room? Have you tried this? There are usually two-way switches, right? You can switch on the light from the entrance and from the bed as well, right? But now I'm only showing you the switch at the entrance. Can you actually tell if the bulb is on or not? Try this, okay? It will not work. The reason is that these are two-way switches, and it is the state of the two switches that determine whether the bulb is on or not. It always has to be 0, 1. Right? It can't be 1, 1 or 1, 0, 0. So that's the one that determines whether the bulb is on. But what is this? This, when you are looking at one switch, this is partial sampling. You have a two-variable system, you're only observing one of them. And partial sampling is actually a very good algorithm to generate random numbers. Even if the calculation is deterministic like this one, so this is for Neumann, he developed this algorithm, the first random number generator algorithm. The idea was that take a 3-digit number, square it, keep middle 3 digits, square it, and so on. And now his number sequence is pretty random. You can have periodicity in this, so it's not very good, but it's still a good enough random number generator. So what we are doing with our measurements when we looked at MEG data, there are so many variables that determine the brain state, healthy or unhealthy, and we picked up one. And we are hoping that this is what will give me a classification. But what I actually have picked up is a random number generator. It's in an amazingly lucky scenario that this should work, or for the patient, amazingly unlucky situation that this thing works. So that's one problem, that partial sampling, that why this will not work, why it's not a data problem, in the sense that you don't need more data or good quality data. Perhaps what you need is more kinds of data, multidimensional. Okay, what's the second problem? And this is a thought experiment that first I want to show you using the modern lingo of RNNs. So let's say that I have two networks. They're identical in their structure, and I'm going to train them for an identical task, like some trajectory they have to match. So this is an easy problem. I can train this. However, my training algorithm has some stochasticity. That means that these two networks will turn out to be slightly different from each other, but not drastically. And if now I imagine that this is a person 1 and person 2, if you look at these two persons behaving, meaning predicting this task, you'll say they're perfectly healthy. And indeed, so blue is my true trajectory that I want this network to follow. And orange and the green dots are the ones how these two RNNs are behaving. So this is all a nice exercise in machine learning 101 nowadays. So then we do this following. I change my setup. I take the output of the first RNN, which we have argued is healthy. And now I drive the readout of the second neuron. Okay, so I rewire from here to here. Basically, I do a brain swap, if you wish. I take brain activity from one person, put it into another person. What will this person behave like? So these RNNs is just a example. Okay, don't take it literally. But in this, what happens is that these red crosses are the output when I swap the output. So this activity in this RNN1 and RNN2 in our comparison is perfectly healthy activity. However, when I mix them up, it's perfectly unhealthy activity. Okay? So what I want to argue there is that there's no real definition of what is a healthy activity. So this brain swap idea, that if I can swap two healthy brains and observe their readout, they both will look sick. However, the activity was actually perfectly healthy. And this partly relates to the fact that the whole brain decides the function. I do a sleight of hand here because what I'm doing is I'm keeping the readout same. Right? The whole system was working fine. Partial system is not working fine. So the partial sampling problem, which I called random number generator, is actually having even more serious problems. That if you did this, it will not work. But more importantly, this is telling us that, oh, by the way, before I do that, this idea, I built it around brain activity. But this same thing applies to, let's say, gene markers. It applies to the network structure. It applies to the chemical makeup of the brain and whatnot. So individually, the argument applies not just to the brain activity, but to anything. So any biomarker search with this approach is doomed, basically. So I don't believe that biomarker search is doomed, but I think our fundamental approach is doomed. So this cohort comparisons that we are doing, that does not make any sense. Because every individual is unique in some way. So that's the second problem. And I think this brain swap very neatly illustrates the problem of cohort comparisons. The third problem, so some friends, they wrote an article, it's a summary of a symposium they had at SFN, and they titled it as Brain's Best Kept Secret. What is the best kept secret of the brain? That there is degeneracy in the system. Degeneracy not in the disease sense, but in the functional sense that one structure can produce many different kind of behaviors, and many structures can produce the same. So that kind of degeneracy, that same components can give rise to different function and vice versa. They illustrated that this exists not just at the molecule levels, same kind of structures could appear in different membranes and in circuits and networks of networks and whatnot. And this is exactly what becomes a problem now for the diagnosis. So what happens? You go to the doctor with these symptoms and they try to pull them together into a diagnosis. If you go back, there are multiple causes for these symptoms. If you go back further, there could be many more and so on. So there is this convergent, divergent relationships between readouts at any given level of the brain and eventual behavioral problems. Moreover, as I earlier was saying, that it's not that just because you have synchrony increase in your system that you end up with epilepsy or something like that. It's not one change, it's many places simultaneous. So that's also makes it very difficult. Now, it is true that there is degeneracy, but it's not completely true because these variables that define my system behavior, they may be correlated. Meaning any arbitrary combination will not give me the desired readout or desired state. So there may be like different manifolds in which you can get similar kind of behaviors in the parameter space. And that kind of gives us some hope that maybe partial sampling is not so bad and sometimes it can actually work. But this also tells us that reading multidimensional biomarkers is actually maybe a hope, because individual readouts may not give you, but their covariances could give you a better separation between the patient. And this again asks for not just partial sampling, we need to read out many different variables. So this approach that we are locked into right now, I don't think it can work. And I can say this in this crowd because there are not enough doctors to attack me, because I'll be more careful if I say this in the hospital. But here, I don't think it will work, because we can only do partial sampling and there's no definition of healthy control, and complex systems have degeneracy. So that's the gloomy message, that all these repositories that exist, they're hopeless. They're never going to work. This is a good playground for our students who are trying to learn machine learning. They have rich data. They can, learn machine learning, but it's not going to solve our problems. Okay, so if you want to comment at this point or questions. Convinced? Yeah.
Are you suggesting we should develop multimodal models, or?
That's what I will try to argue here a little bit more. But at least current state is the following, that these repositories which give us this illusion that we have now a lot of data and we should be able to help with the diagnosis at least. I don't think this will work at all. Maybe in very serious cases of epilepsy or Parkinson's disease or Alzheimer's, but that's not the point. I mean, there you can do the diagnosis on behavior. Why do you have to go all the way to the brain? And you can't treat them anyways. Okay. Then what is the future? What can we do? This is my second part, maybe a little bit hopeful. Something must change. I think we have to get out of the old plan. Now, in today's time, whenever you feel these kind of problems, your immediate reaction is that, but there is AI coming. That will help. So yes, indeed, AI will help us. It will help us in collecting data, organizing data, and we are almost doomed to succeed in some sense, meaning you will put a lot of data into these AI models and they will give you some classification. But is that useful? Because most of this will be based on correlations. And a doctor perhaps is not interested in correlation. If they want to treat you, they want to know some causal links. So what is difficult and where AI may not be that effective is defining what to measure and how to go from correlations to causations. And I think a way out is that we have to complement our AI-based descriptions, this data analysis of multimodal data, etc. We have to combine it with the more mechanistic, or let's say biophysical models of the brain. And that's the only way I can think of to link measurements to biology. And that's where things will start to make sense. I'll explain what that means. But other thing that has to change is that we have to get out of this cohort problems, comparisons. When you go to a doctor, you never measure the distribution of that particular readout in Stockholm or in Sweden and say, oh, today I'm 2 standard deviations out, I should see the doctor. You actually compare yourself with your previous self. Like you have some sense, I've been sleeping okay, but last 2 weeks or so, my sleep is getting disturbed. I should maybe go for a sleep check. Right? So we compare ourselves to ourselves and then we go to the doctor. So I think that's the first thing that should come in, that we should create means to do this first-person readout of the brain diseases. And here I want to give another example from Pascal's work only. So we took that data. We hadn't given up hopes until that point. So we said, no, our method is not good. So the spectral analysis is not rich enough. We should do something more. So I met a friend at Norita, and he had some ideas from non-equilibrium physics. And he said, how about we try that? Okay, That makes sense. And we also cooked up a theory that how brain structure should be related to this non-equilibrium measure called entropy production rate. What it really measures is that if your forward trajectory is equally likely to the backward trajectory. So if your system is more like reproducible in this sense, or it has more random repertoire. So we measured this from the same data that Pascal had analyzed earlier, and this is what it looks like. So each line is an individual patient, and the colors tell you whether they belong to healthy controls or Parkinson's disease on or off medication. So this is, the thick lines are the averages. It's nothing here. Right? It's basically useless. But good exercise. We developed the algorithm, it can be used further. However, Pascal still had hopes. So what he did was that he compared this entropy production rate of a patient with itself. And in this case, we could do that because we had measured same patient, same healthy control twice, and same patient twice, once with medication, once without medication. And these arrows that you see are the individual patient arrows. They go from unhealthy, so off medication, to on medication. And now you start to see that some subjects, some patients who really had very high entropy production in their off state, after taking medication, it actually comes down. And it tries to go towards this circle where most of the arrows belong to the healthy control. So in healthy control, of course, this measure fluctuates, but it remains mostly in this gray oval. So this kind of says that we should not do this. Actually, we could compare a patient to itself. So in this case, it was good that there was a pairwise comparison possible.
Can you just define this entropy production you talk about? What do you mean by that?
Oh, entropy production, the idea is that, let's say that I have a discrete system which has 3 states. Now, probability of going from I to J and J to I, are they same or different? So if your system is in equilibrium, the probability to go from I to J is same as J to I. However, if the system is in non-equilibrium, or let's say it's a neural network which has some structured connectivity, so the moment you give arrows to your synapses, you're going to change it. That if it is from I to J, then of course in this 2-state system, there's no transition from J to I, unless this is spontaneous one. So we compare these probabilities that if forward transitions are equally likely as the backward transitions. And the reason it is interesting was that, of course, dopamine is involved, and dopamine affects synapses. So actually, the initial hypothesis was that lack of dopamine will make the system more equilibrium-like. But it turned out it actually pushed them out even further from it. And so much for hypothesis-driven research. Okay, good. So yeah, what Pascal argued here is that actually it makes more sense. And this is not unlikely. I mean, this is if you dig into literature, you will find a lot of this kind of pairwise comparisons, which makes more sense. So that's what we concluded, that we can only compare individuals with themselves. Here is my slogan. A good model of a patient is a patient, preferably the same patient. Okay, and this again asks for the same individual-based classification of symptoms, etc. So what we need for this is that we need, if we want to compare a person to itself, what we need is regular measurements, cheap measurements, reliable measurements, objective measurements. You can't go to a clinic every day or every week to get your brain scan so that you know that if Alzheimer's is coming or not. That's not going to happen. So we need something different. And I think that's where the technology plays a role. Heart measurements were extremely rare before the Fitbit came in. So somebody made this Fitbit idea, put it in the watch. Now everybody is measuring their heartbeat and pressure and so on. You could question, is it really good to have such over measurement? But I think it's useful in this sense that if you have good algorithms now, which keep track of these variables and then could flag you something. So that's the first future I would like to think, that you need these means to measure and share data in a better way. And that's where digital future comes. Second future is, as earlier I alluded, that AI alone is not going to be very useful. It has to be complemented with brain models and most likely personalized brain models. So you get a lot of these biomarker data. This is valuable. We pass it through the pipeline of AI as well as our more biophysical models. And then you tell the doctors that, hey, this is the extra behavior or extra problems that you might have ignored when you were just looking at, let's say, brain morphology or just looking at the dopamine degeneration. So this is a big plan, of course. However, I think it has to be done like this. And this perhaps will remind us of digital twins. So I get a bit upset when people use this term digital twin because I think it's just good old computational models done properly. However, it turned out when I was looking for this, that this notion came up in NASA. And they were building these models so that they don't have to work with the actual physical model. So for them, there was a physical model and there was a virtual model. And they had a data pipeline. So these three things make them a digital twin, so to say. And earlier they were using other definitions. But somehow this word caught on now recently. But still what it says is that a digital twin is an integrated multi-physics, multi-scale probabilistic simulation of an as-built system enabled by a digital thread that uses the best available models, sensor information, and input data to mirror and predict activities' performances over the life of its corresponding physical twin. And then I was remembering this line from Lewis Carroll, that only satisfactory map to scale of a given country was the country itself. Anyway, so for the brain, can we actually do this? Is it really possible to go to this level that you can have all integration of different physics, different scales, etc.? For a car, maybe possible. Maybe possible for a rocket, perhaps possible even for a nuclear bomb, because all the physics is understood. But for the brain, we are still discussing what is a good readout of the brain. So I am not so sure whether we can directly jump to the digital twin. However, what is possible is local solutions. That you can now, through this biomarker search or through the computational models assisted by AI, those possibly could work as not a twin, but sort of a replica of a specific aspect of your brain function. And that may be useful. So I don't think we should oversell this because it has a lot of potential. And if we oversell the idea, we basically lose it. We can't make twins, but we can make reasonable copies. And how will they help? They will help, first of all, as I earlier said, that we have multiple features and they are correlated perhaps. But what possibly happens in diseases is that these correlations could break. And each patient could follow its own trajectory in terms of how they are going. So where these models could help is actually sorting out these trajectories into different groups. That all these patients or healthy group, perhaps they look alike with small deviations, but one of them evolved into this category, other evolved into this, and so on. So they look different after they have had some perturbation, some changes. And this is where I think the role of models come in separating these, let's say, growth, evolutionary trajectories of the brain. And in this big scheme, that's how it looks like, that you have epilepsy. It can have many symptoms, and that could have many other relations to the brain activity and so on. So each person will be living on one of these trajectories. And that's the sorting task that we need to do using models. And for that, it is not sufficient to just focus on brain activity or symptoms or genes, or you have to have at least few readouts simultaneously from one person to narrow down whether the person is living on this one pink trajectory or the other pink trajectory. Of course, there's convergence. But how do you separate between, let's say, the blue and the black trajectories which belong to the same diagnosis, that is epilepsy? They may be differing here, they may be partially overlapping here, they may be totally converging here. So that's where I think models should help us to find right trajectory for each subject. So that's one. And of course, this requires that we ask very fundamental questions, and those fundamental questions are relating to how to bridge these scales. This is also hopeless to say that I can directly jump from here to the whole brain. I need to find bridges. So that's, I think, a very, very basic science question. The other thing that we need to do is basically change gears. And I want to go back in 1961 when cybernetics book came up. And in cybernetics book, there's a very nice line. So first of all, he said in very nice words that psychopathology has been a disappointment. It was a disappointment then, and I think it's a disappointment now. At that time, Norbert Wiener said that it is not surprising in considering that functional mental disorders as fundamentally diseases of memory, of circulating information kept by the brain in active state, and of the long-term permeability of synapses. So Norbert Wiener already at that time is creating a term called synaptopathology, or something like that exists, of information, diseases of information processing, diseases of dynamics, diseases of memory. He's not talking about diseases of epilepsy type. He's saying that that should be a disease of stability. And so I think what we need to do is that perhaps change the perspective once on how we define diseases. Basically, from a clinical perspective, you need to worry about symptoms because they have to be managed. But in terms of understanding the disease, finding the right therapeutic trajectory, we need to change our perspective. How would it look like? In my thinking, it is how it will be that you have this definition of the brain. So the definition of brain should lead to one set of classification based on just the dynamics of the disease. And I'll give you some examples of that. And similarly, if you treat it as a problem of information processing, then there should be diseases of information processing. And I hope that this will give us somehow better effective theories and maybe better biomarkers. So one idea that is gaining some traction, especially in the field called computational psychiatry, it starts with some very influential thoughts from David Marr. David Marr very nicely articulated an idea that perhaps was already there, that how do you understand an unknown system, especially that of a brain type. So he said that you have to define what are the computational problems this system is solving. He took the example of vision, but you can think of a motor cortex or something. And then you ask, what are the possible algorithms to do that? So if I want to, let's say, detect edges in this environment, how do I do that? So edge detection is the computational problem. Then what are the algorithms to do that? And once I have my catalog of algos, then I can ask, how can I implement them given my hardware? So this is how David Marr proposed how we should approach the brain science. So now some people take this idea more seriously and elaborate it in a beautiful way, I think. So they think of it in terms of diseases, especially psychiatric diseases. So you could say that, hey, I have some psychiatric problem. What is the computational level of this problem? Is it a problem of, for instance, exploration versus exploitation? Is it a problem of stability versus variability? Behaviorally, this is a balance I need to figure out somehow. How do I explore? When do I exploit? Whether I remain stable in my choices, whether I change it. Now, once you know this, then you could say what possibly could be wrong at the algorithmic level. Maybe it's a change in the stochasticity of the neurons. And then you can ask, how can I do this? How was it implemented? Well, stochasticity could be implemented in the gain of the neurons. So this way of thinking gives you a different entry point into this problem. And not only that, you can think about it like this, you can, based on this, you can define your measurements. So if you are talking about just computational problem, it makes sense that you read the behavior. But if you are talking about the algorithmic level, then perhaps you should look into brain activity. And if you are looking into the implementation level, then you should really cut into the brain and find out what's wrong with the synapses, neurons, etc. And same thing in terms of the therapies then, that if it's a behavioral problem, of course CBT could be helpful. However, if the problem is here, you can talk endlessly to this person, nothing will happen. right? If the problem is here, you have to take medication. So I think this gives a very nice perspective, and I believe that this should be part of our digital future of brain diseases, that we start to think diseases in this computational. And there is already something going on in this. For instance, you can think of ADHD as too much exploration, addiction as too much model-free behavior, schizophrenia more top-down reliance as opposed to autism, which is more bottom-up driven brain processing. So already some indications are coming up, but they have to be scaled up to the level of clinical viability. And the second thing is about dynamics. So under this information processing system lies the dynamics of the brain, and of course that can go wrong. This dynamics we can abstractly represent, but we can also observe it in terms of neural activity and behavior. For instance, this is data from locusts, where if you give them different inputs, activity goes from some baseline to a fixed point, which belongs to that particular stimulus. So if you treat the brain as a dynamical system, then immediately you could say that changes in the brain state are changes in its stability landscape, the kind of fixed points you have, some bifurcations may have happened, the stability of individual fixed points, that region of convergence may have changed. And that would be another perspective to diagnose. And obviously, when I'm saying dynamics, I'm not referring only to the dynamics of, let's say, brain activity. It could be, let's say, the interactions of the genes. So the gene interaction networks could also be thought of as dynamical systems. So here is just some example to wrap up quickly, is that like epilepsy, for instance. So this is an animal model of epilepsy where we have created epilepsy of a particular type. And you look at the brain activity, just by looking at it, there's no way you could determine if something is changing here, and that is leading to. But obviously, if you think of it as a dynamical system, possibly you can start to see, like, is something happening to the stability landscape? That maybe now the stability of this fixed point has changed so that there is a probability, higher probability to make a transition to unstable. Similarly, you can think of Parkinson's disease, where there is a characteristic oscillation that is observed in the basal ganglia, which could be like a change from a fixed point to a more limit cycle case. And this is another beautiful example where the brain loses the ability to synchronize oscillations. It can produce oscillations, but it can't synchronize them. So these are problems of dynamics. And this is something that I was quite surprised to find when at some point I looked at, let's say, oscillopathies, if you wish. So here's a disease called Parkinson's disease. You have 20 hertz rhythm, very strong in basal ganglia. Here is epilepsy example. Epilepsy in absence, meaning these guys, they suddenly blank out. And in their thalamus, there is same kind of rhythm that is seen in Parkinson's disease. Just that basal ganglia happens to be the motor output, so Parkinson patients have problem in expressing their motor repertoire, and in this case, it's in the sensory input side, so nothing goes in the brain. And this is, of course, as I earlier said, that this is a problem of synchronization, that schizophrenic patients, they have problems in synchronizing their oscillations. So in a way, there are different symptoms, different diseases, different changes in the brain, but at one level, some invariance appears, and that invariance is in the form of oscillations. Whether they exist too much, if they exist, whether they synchronize. So I think this gives a very neat perspective to rethink about the disease conditions, and possibly gives us better means to intervene. Like, for instance, in this case, it's clear what you need to do then. That if schizophrenia is defined to whatever extent by this readout, then you can alter it. There are means now. Or in this case, it was much easier that you stimulate the brain so that these rhythms disappear. And this is best illustrated in Parkinson's disease in deep brain stimulation, that you simply abolish these rhythms by whatever means. And a lot of symptoms disappear.
The motor ones.
The motor ones, exactly. Yeah, but also, I think motor ones disappear because only then the patients show up in the clinic.
That's the problem.
I think early, if it could be done very early on, perhaps. Anyway, but you're right, only motor ones. So yeah, that's, I think, the perspective from the dynamic side. So to summarize what I said is that I think the current approach is not going to lead us further. It has brought us wherever it could. More repositories, better algorithms will not help us. We need to change our approach. And I think that approach should be, first of all, focus on individuals. And that means making more measurements of different kinds from the same individual. And on the other hand, we need to change even our viewpoint on the diseases and be more computational or more physics-based to ask these questions. Yeah, thank you so much.